Hey everyone, Mr. Murphy here. Let's talk about Chapter 5.5, which is all about the response to environmental signals. A signal transduction pathway is a complicated mouthful just to say it's the series of events that occurs from a signal to an event. And that can happen for a variety of different reasons. There's lots of different types of signals, from response mechanisms to like something that startled you, or you're getting older and hormones, or whatever. So the different types are autocrine, which are signals that affect the same cells that release them. So that's a signal that is affecting the cell itself that sent that signal out. Paracrine, which diffuse and affect nearby cells, and then hormones are just a broad term that we use to refer to signaling molecules that travel distances, usually through the bloodstream or the lymphatic system or something along those lines, in order to reach distant cells where they are able to cause their activity. Here's a diagram showing you kind of all three. A secreting cell releasing signals out into the extracellular matrix. That signal could be autocrine and come right back to it. It could be paracrine and go to nearby cells, or it could be a hormone that ends up in the bloodstream and then lands on a target cell, which might be very far away. So cells that have the receptors to receive these signals are the ones that are going to respond to them, and not other cells. So that's an important thing to understand. Those signals that go out, they need to land somewhere and cause a change. If a cell doesn't have the necessary proteins either on its surface or inside of it in order to do anything with that signal, then nothing happens. So here's an example. We have a receptor, which is just a protein that's hanging out probably in the cell membrane. It could be a cytoplasmic receptor. So there are circumstances where hormones might be lipid-based, where they can actually sneak right through the cell membrane, but they still need to be able to hit that receptor. And the most typical examples that we're going to go through in this chapter are cell membrane bound receptors. So here we have a receptor. It receives its signal molecule. Because chemistry, that signal molecule is going to cause a change in the shape of the receptor, and now that that receptor has a new shape, its activity may be activated in some way where it then changes some other protein into an active shape. It then changes multiple other proteins into an active shape, and it can be this whole cascade of events where ultimately it leads to whatever the intended action is. So maybe that could change the rate of transcription in order to cause the cell to start producing things. Maybe it activates other enzymes inside of that cell to again, facilitate transcription, to facilitate the production of proteins, to shut down either of those events. It could be any number of things. This is a example of allosteric regulation, because you notice this signal molecule landed on the receptor. It never did anything else. At that point, it's changing the shape of the receptor, which then changes the shape of its active site and enables it to then do its job. But the signal molecule is not actually involved in that. So the signal molecule is just changing the shape of another protein, which is what we saw similar to, or what we saw as an example with enzymes being inhibited through allosteric sites. Here we see that these signal molecules can activate proteins in that same way. So the ligand, recall, is the word for basically a signal molecule that's being received by a protein. It's got a three-dimensional shape that causes a shape change. I feel like I'm beating a dead horse by talking about this all the time, but it's the chemistry of it, that this molecule has a particular push and pull because of hydrogen bonding, which is going to change the shape of this. I like this diagram because it's like, look at us, we're drawing these like lumpy proteins, and then it's just this like weird little ball thing here. That's not to say that's not what the protein exactly looks like. It's just kind of goofy to think that biology is weird. So here this ligand lands. That's going to cause a change inside of the cell, which can facilitate then the activity that we intended. Now, they're not usually like metabolized or broken down or anything, these signaling molecules, and they're subject to their own diffusion and their own interaction with the extracellular chemistry of the cell. And so basically it's a game of concentrations. And we've heard about this before, but it's good to keep in mind that inside of the cell, it's never black and white. It's never just like you have the molecule or you don't. It's, do you have enough of the molecule to, in general, be causing the event that that molecule causes, or do you not have a high enough concentration, and so in general the event is not happening. It's never just like a straight on or off. It's just, have we reached the threshold where enough of it's happening, or have we not? Binding, therefore, is generally reversible. It could be released, and it could bounce back if the concentration's high enough. It could be going either direction. There's also inhibitors that can block that. And a lot of times, the body has its own inhibitors to control these mechanisms, but also a lot of different types of pharmaceuticals and things are all about blocking these receptor sites in order to maybe inhibit pain reception or things of that nature, to sort of step in and control something that might otherwise be causing problems in a person. But of course, you can understand then how that's a pretty high stakes game of trying to make sure that the concentration is right to inhibit the bad side of what you're trying to prevent, while not shutting down the ability of the cell altogether. Turns out that coming up with pharmaceuticals is kind of hard. Who knew? So these receptors are classified by their location within the cell. So, and it depends on whether the ligand can make it through the membrane. I mentioned that before. So cytoplasmic receptors are in the cytoplasm, and that's assuming that the ligand has made it through the membrane, either because of a channel protein or some sort of facilitating protein, or just because it can pass straight through. Maybe it's really small or nonpolar, it just sneaks right in. Membrane receptors are ones that are on the outside of the cell, and that's what I was referring to with the diagrams before. They're also classified by their activity. So we have ion channel receptors, protein kinase receptors, or G protein-linked receptors. The ion channel ones are pretty simple. These are the gated ions we saw earlier in the chapter. Basically, something lands, opens a channel, allows ions to move through. It's kind of a very deliberate and quick action then to get from the signal to the intended consequence of an opening or a closing of a channel. Protein kinase receptors are when the protein that the ligand has landed on changes its shape. We saw that in the previous diagram, or earlier diagram, and we'll see another example of that shortly. But in this case, then, that changed shape now leads to a new activity for that protein, which then leads to something else happening, and something else, and eventually gets its way to the intended consequence. So here, a signal lands, in this case insulin. The insulin causes this protein kinase to change its shape. It becomes bent, which then facilitates the hydrolysis of ATP, which allows further for this shape change to interact with a target cell. That target cell gains the little phosphate group and then does its thing. I don't want to burden you with following phosphate groups and things, but if I threw an example like this at you, what I want you to understand for a protein kinase is that the signal lands, causes a shape change, and ATP then facilitates. It's an energy requiring process that facilitates stuff happening inside of the cell. And that seems vague and kind of lame, but honestly, we're learning this at a level where we're trying to understand the broad strokes of how this signaling works. And it's sort of a waste of our time to be hyper-focused on very specific examples. Because at the end of the day, it's the same basic principles always going on. It's just sort of we're tweaking the fine details of exactly what molecule is being moved or changed or whatever. So here's basically this reaction. ATP plus the protein leads to the ATP being broken down into ADP, and now a protein has been phosphorylated. When you attach a little phosphate group, that changes the shape of this thing, which activates it. And what's crazy and kind of mind-blowing, if we're thinking about ATP in the traditional way, it's the energy carrier of the cell. Actually, it just allows for phosphates to be attached to proteins. And when a phosphate gets attached to a protein changes its shape, and that change in shape can do stuff. That's the function of ATP. It's not this magic I grant upon you energy to do stuff. Instead, it's just, you have a new shape, you do new stuff now. And that's how it drives it forward. Now, it produces energetically favorable conditions for the further actions of the protein to occur, but can be a valuable thing to sort of start rethinking what ATP really is. It's a shape-changing molecule. Not that it itself is changing its shape, but that it facilitates the shape change of other molecules. So protein kinases work in that way, that you have an ATP and you've got a protein, and when the signal lands, that ATP causes a shape change in some target, which then causes further things to happen inside of the cell. The other example is G protein-linked receptors, and this seems kind of confusing. It's the exact same thing we just saw, just sort of mediated with an in-between protein. And so in this case, you have the receptor receiving the signal. You have GDP and GTP, just guanine instead of adenine. It's the same basic mechanism of energy transfer occurring. So GTP is an energy carrier in the cell. It's just like the less famous cousin of ATP, but it's still very important inside of the cell. Just don't get bogged down by like, oh my gosh, GTP or ATP. I'm not going to worry about the specific energy carrier. I mean, in the case of G proteins, yeah, okay, maybe we should remember it's GTP because it's in the name there. But the point is, it's just an energy-carrying molecule. And then you're going to have an effector protein that's going to be stimulated by this process. This is most easily understood just by looking at the diagram. So here you go. We have a signal, hormone, lands on the receptor, and where the protein kinase would have changed the shape of this thing to directly lead to the internal cellular action, the G protein-linked receptor has a little G protein that it's linked to, either in proximity or whatever. But what's happening is when the signal's received, it leads to a change in the G protein, which leads to the phosphorylizing of this GDP into GTP, which then changes the shape of the G protein, which then you see this little subunit breaks off, goes over here, changes the shape of this effector protein, which then can do the job. So if we put the two next to each other, the only difference is that the G protein linked sort of has this extra step of this G protein that needs to be activated by the receptor in order to then activate the next protein that leads ultimately to the product. So that's really the only difference. It's otherwise very similar to the protein kinase example that we saw. Short version of all of this is signals received, stuff happens, proteins change their shape, you get the target action inside of the cell. And that is chapter 5.4, which is a lot to wrap your head around, so definitely ask me questions if you have them. Thanks.